Greetings everyone and welcome to another Genetic Engineering and Biotechnology News webinar. Our presentation today is Next Generation Phenotypic Screening, enabling miniaturized physiologically relevant assays at fast speeds in precision medicine and beyond. I'm Jeff Bogaliskis, Technical Editor for GEN, and I'll be the moderator for today's webinar presentation. In the drug discovery world, speed is king. Biopharmaceutical companies are continually searching for unique methods of leveraging automation with biochemical assays that can quickly assess thousands of potential drug compounds. New high-throughput screening devices, such as the IntelliCyt iQue screener, can generate high-content data at speeds on heterogeneous cell populations using miniaturized cellular assays or in a multiplex, ultra-miniaturized bead-based assay format. Data from these devices should allow researchers to perform more physiologically relevant experiments faster, while conserving precious samples, saving costs, and overcoming the limitations of traditional approaches, thus paving the way for faster development of therapies and personalized medicines. Let's meet our speakers for this GEN webinar, who will discuss their cutting-edge research and how their methodologies have been streamlined with the use of the iQue screener. Our first speaker will be Caroline Heckman, a group leader and principal investigator at the Institute for Molecular Medicine, Finland, within the University of Helsinki. Dr. Heckman will inform us on her use of multi-parameter analysis techniques in her study of blood cancers. Dr. Heckman will be followed by Catherine Bardell, a senior scientist with AstraZeneca. Dr. Bardell will tell us how the iQue screener has assisted her work with multiplex cell-based assays in her research on ALS. Before we get started with Caroline's presentation, I want to encourage everyone to submit questions for our Q&A session at the end of the presentations. We'll try to answer as many questions as we can, so simply type your question into the Q&A box on the left-hand side of your screen and hit submit. Okay, with all the particulars out of the way, let's get our webinar started. Caroline, the audience is listening.
Hi, everyone, and thanks for the invitation to speak today on this really, I think, fascinating and applicable topic especially for precision medicine and cancer therapy. So I'm going to talk today about an aspect of a very large collaborative project that we have ongoing at the Institute for Molecular Medicine in Finland. So this project is something we started several years ago. It involves several different research groups as well as different hospitals here in Finland and also in the Nordic region. And the idea behind this project was to set up a broad-scale systems medicine study for blood cancer patients. And the backbone for the study was really establishing a very high-quality biobank for hematology patients. With this biobank, we would collect samples at different stages of the patient's disease. So when the disease was first presented, at diagnosis, also after treatment response, and also when the disease would recur. And because Finland is such a small country, there's only 5.5 million people, we wanted to set up a national biobank and also set it up in such a way that we would have very useful samples, not only for the standard, say, genomic testing, but also for phenotypic assays, which I'll describe in the subsequent slides. So as part of this study, we would collect these samples and then we would profile them. So we would our plan was to use a high-throughput ex vivo drug testing assay, which I'll describe, and at the same time do next-generation sequence analysis. So this would include exome and RNA sequencing. And at the same time, we would feed back the information that we would get from these assays to the clinics. And especially for relapsed patients, they could use that information to decide on the next stages of treatment. But at the same time, as basic researchers, we would use the information to better understand the mechanisms driving the disease. So understanding the disease initiation processes and also disease progression and drug-resistant mechanisms. One aspect of this study, and I would say it's a really unique aspect in a precision medicine setting, is our ex vivo drug sensitivity assay. And this was set up here at our institute, FIMM, in coordination with our high-throughput biomedicine unit, and which has the capabilities and the instrumentation to do high-throughput screening. The unit itself was actually set up to do genome-wide screening, specifically siRNA screens, but eventually it got repurposed to do high-throughput drug testing analysis. And in this study, we have established an oncology drug collection, and these are drugs that are all commercially available, and at the present time we have a collection of 525 different drugs. And we prefer to use these commercially available drugs so we can avoid signing an individual MTA agreements with each company. And at the same time, we have some instruments such as the LabSite Access Workstation and use that technology to pre-plate the drugs on 384-well plates in 5 different concentrations in a 10,000-fold concentration range. And then once we get a sample from the hospital, we isolate the tumor cells from the sample, and we can just pull out the prepared drug plates and add the cells to those plates and then start the incubation process with the drugs. So in our standard assay, we incubate for 3 days, and then we add different assay reagents to detect the viability of the cells. And since we're testing the drugs at 5 different concentrations, we can get dose-response curves for each of those 525 different drugs. And we have a very nice bioinformatic pipeline so that we can get the results very quickly and return those results to the clinic. And so we can, with this functional phenotypic assay, not only are we providing useful information for the clinic, but we're also getting a better understanding of disease processes and also looking for novel drug repositioning opportunities as well. And the reason why we can understand a bit more about the phenotype of these cells from this drug testing assay is that our oncology compound collection is composed of several different signal transduction inhibitors. So this slide shows just a part of these inhibitors that we have in our collection. Many of them are already approved clinical drugs, so 28% of our collection is approved. And these can ideally be used for the treatment of these patients. The other 72% of this collection is still investigational. But overall, the data that we get from this analysis provides us with a better understanding of what signaling pathways are active in the patient's cells. So we get hundreds of different dose-response curves for each patient sample. But overall, this gives us a huge amount of data, phenotypic data, to begin to work with. And because it's a huge amount of data, we need to prioritize and make this much more interpretable, especially if we return this data back to the clinic. So for our hematology studies, especially with the acute leukemias, we've developed what we call a drug sensitivity score, DSS. And the higher the DSS value, the higher the sensitivity of the cells to the compound. At the same time, we also screen normal cells, so in the case of leukemia patients, normal bone marrow cells. And we have the drug sensitivity results from those samples as well. And so we can prioritize then the drugs based on how specific they are for the tumor samples. So the higher the sensitivity of the tumor cells to the drugs and compared to the normal cells, then the better the response or select response of the tumor cell to that particular drug. But if the normal cell sensitivity is greater than the tumor cell sensitivity, then we know that we probably want to avoid those drugs. So this type of information we can feed back to the clinic, and they can implement it, especially for patients that might be relapsing from off of the standard clinical drugs that they're using. So in these cell-based assays, and especially this first generation assay that we have implemented in our program, there are some advantages and also disadvantages. So our assay is based on a reagent that we use called cell titer glow. But the problem using this type of assay is that we can't distinguish between different cell populations that might be making up these heterogeneous patient samples. And this leads to the possibility of misinterpretation of data. However, these assays are very robust, they're very inexpensive, and they're very fast. But if we think about applying a flow cytometry-based method, there are several advantages as well. So because we're studying complex patient samples, we can study, using a flow-based assay, we can study the samples at a single cell level. And so we can avoid doing, say, cell enrichment, and we can look at several different cell types at the same time. And using, for example, a high-throughput instrument like the iQue Plus from IntelliCyt, which has 3 lasers and 15 detection channels, we can get a huge amount of information from very heterogeneous samples. Applying high-throughput flow is also faster, and because the sample size requirement is smaller, and also the assay size is smaller, it's less expensive. And we can, at the same time, produce a huge amount of data compared to maybe a lower throughput standard flow cytometry instrument. And then just some points about studying and applying these methods to, say, primary blood cancer patient samples. The reason why one of the reasons why we started off studying these diseases is because the samples were easily accessible. And we could apply the assays that we are using on peripheral blood or bone marrow aspirates taken from these blood cancer patients. For acute leukemia patients, the tumor cell content can be easily monitored by clinical tests, and this is usually provided as a blast %. But the samples are extremely heterogeneous between patients and also between sampling times. And they're also, in terms of heterogeneity, complex in the form of being composed of many different cell types. And therefore, as I had mentioned earlier, the single parameter assays are quite limited in what type of information you can get and interpret from these samples. In addition, single parameter assays don't really give you information about the impact of the tumor microenvironment. And also, if you're looking at drugs that target the cell proliferation capacity, these primary cells usually don't proliferate very well ex vivo, and these single parameter assays, therefore, are also quite limited in trying to read out the proliferation of the sample. But if we move to multi-parameter assay and try to understand the drug effects on these patient samples ex vivo, using instruments such as the iQue screener, we can easily identify the different cell types that might be affected by these drugs. We can also look at different functional readouts, proliferation, cell cycle, apoptosis, as well as cell death, protein phosphorylation, and also different aspects of the microenvironment, such as cytokines that might be present in the sample. We also have no need for an extra control because there is actually an internal control. We can use the normal cells that we can detect based on the cell surface staining. And it also would allow for us to study samples with very low tumor content. So single parameter assays, we really can't, we're just getting one readout. So if there is a sample with a low tumor content, then the readout that we will get is primarily from cells that are not tumor cells. And also, if we apply high-throughput analysis and combine it with a multi-parameter assay, we can measure multiple drug concentrations, so we can get those dose-response curves as we did earlier with the single parameter assay, and we can also measure the samples at multiple time points. And because we can use a high-throughput assay, we can reduce the cost of the assay, and we can also reduce the requirement of the sample size as well. So the first project that we started on was focused on a disease called acute myeloid leukemia, or AML. And this is the most common acute leukemia in adults. Patients that are diagnosed with this disease have very poor survival. It's extremely heterogeneous, immunologically and also genomically. The therapy for these patients really has not changed in the past 30 years. And there are no real good targeted therapeutic strategies for this patient. And also, if we look at the survival curve shown in the lower right graph, we can see that once patients relapse, their outcome is extremely poor. So, and basically, there are not very good treatment options for these relapsed patients. So there is really a great need to find new therapies, especially for relapsed AML patients. So much of our analyses are based on getting and applying our assays to AML patient samples. And this slide is just showing the process, the steps involved in getting a sample from the clinic and just setting up a high-throughput phenotypic assay. Basically, this does not take too long. Parts of it we do have automated, but basically we get a bone marrow or blood sample from the clinic. We do a gradient separation to isolate the mononuclear cells and get rid of the red cells in the sample. We have our pre-drugged plates that have already been prepared, and so we just need to add the cells to those plates. And depending on the drugs, we incubate for a certain amount of time, so either from 1 to 3 days. And then afterwards, we just pellet the cells gently, get rid of the supernatant, add the antibody and reagent mix, incubate briefly, and then analyze the plate on the iQue screener that we have. We actually have two of these instruments at our institute. We have the iQue HD and we have the iQue Plus screeners, and both of them are quite popular and in very high demand by many of the researchers here. So for AML, we can then set up different gating strategies depending on what cell subpopulation we're most interested in studying. We first need to isolate the single cells, also separate out live cells from the dead cells, and then after that we use a CD45 marker to isolate the leukocytes. And then also, depending on the patient and the immunophenotype of the patient, we use different markers to isolate the blasts. So for the example shown here, and I apologize for the resolution of these graphs, but we're using CD34 and CD123 to stain the cells and isolate the AML blasts. From the CD45 positive cells, we can also gate out the stem cells, and also this is based on the immunophenotype of the patient. So in this case, we use CD34 and CD38, and then we can further look at the leukemic stem cell population, which is quite small, but I think one of the problems with AML relapse are the leukemic stem cells. And so there is a very high interest to find out how we can target these cells. So it's important to know how you can gate them and identify them in this assay. But in this case, we were using CD96 for staining. So then, based on that gating strategy, and because we have the drugs plated in several different concentrations, and we might also have different combinations on the plate, we can get a dose-response curve for each of the different populations that we're interested in looking at. And then we can see from this analysis that each of the drugs that we have on that plate will have a different effect on these different populations. And we can see from drug 1, we have a drug that actually highly affects leukemic stem cells, also leukemic blasts, but doesn't affect the normal hematopoietic stem cells as much. We have other cells that might, we have other drugs that might affect normal cells much more than they affect the AML blasts or the leukemic stem cells, like drug number 2, for example. But this is all really a huge leap forward compared to our single parameter assay. So we're getting so much more information with this multi-parameter assay and getting so much information about these drugs and how they're affecting the different cells composing our heterogeneous patient samples. And if we compare the results that we get just from the number of live cells from our iQue instrument compared to a standard single parameter assay, we can see that the results pretty much correspond. There might be, in some cases, some overestimation with the single parameter assay compared to the flow-based assay on the effects of some of these drugs. But basically, there's pretty good correlation. And just a few comments about working with primary samples, especially these blood cancer patient samples. We had been utilizing our biobank samples. We also primarily use fresh bone marrow or blood, but something that we noticed when we started applying the multi-parameter assay to our study was that, well, if we're using frozen bone marrow samples, we're starting to lose certain cell populations, and this is primarily granulocytes. So anybody who is interested in studying granulocytes should probably use, or should just stick to using fresh samples and avoid using frozen samples. But otherwise, most of the other cell populations that we have been interested in stay intact even after they're cryopreserved. And then we have started off using a 96-well assay for the multi-parameter analysis, but we have scaled up to a 384-well format, and the iQue screener process is capable of reading 384-well plates as well as 96-well plates. And we still find very good gating of our different cell populations. So there hasn't been any problem in scaling up to a 384-well format. And just some other interesting observations that we've had from this multi-parameter analysis is that for certain common drugs that are being used in the clinic, for example, this glucocorticoid dexamethasone, what we have realized from our analysis is that dexamethasone is actually very specific for lymphoid cells compared to myeloid cells. When we look at just standard bone marrow, we really don't see too much an effect with dexamethasone. But when we start looking at the different cell populations, we see a distinction between the drug effects and the different cell types. So, for example, dexamethasone is much more specific for B and K cells than other lymphoid cells and, as I mentioned, myeloid cells. And it's also really not very effective for other certain cell types, but really specific for these B lymphocytes and NK cells. And then also another drug that is widely used in research labs nowadays are Bcl-2 inhibitors. In the case of navitoclax, which is a pan-Bcl-2 inhibitor, it's not used in the clinic, but we're interested in using it as a tool in our studies. We also find that this drug is very specific for lymphoid cells compared to myeloid cells. And this is also seen from this dose-response curve. But basically, most lymphocytic cells are sensitive to Bcl-2 inhibitors compared to other cell types. So these are our observations that we're now making because we're transitioning over to a multi-parameter assay, and we're making so many more discoveries with this transition. And I think this is going to be useful not only for our basic research studies, but also for the understanding of the clinical impact of these drugs. So many drugs, for example, fail going through development because of their off-target effects. There isn't really a complete understanding of how the drugs target the non-tumor cells or the healthy cells compared to the understanding of how they target the tumor cells. So I think we need a much better understanding of the whole entire effect of these drugs and not their more specific effect on the tumor cells. And just to wrap up and summarize, I think overall what we're finding from our project and from these assays is that when we can apply ex vivo drug testing analysis to primary patient samples, what we are finding is that it can be very, very helpful for predicting drug efficacy. And our clinical partners are using our results. Basically, often we are not able to find a drug that is helpful for their patients, or it has not been approved, so it's not available. But the results also give a much better idea of drugs that may not be working at all and should be avoided. We can also see from our drug testing assay that we can start predicting patient outcome. I didn't go into any of these results, but there is a correlation between the ex vivo and in vivo results. And we also have a few publications showing some drug repositioning possibilities as well. And then I think I couldn't emphasize enough in this presentation that multi-parameter analysis really allows for a multifunctional readout, and we can look at the effects of drugs on several cell types simultaneously from very heterogeneous samples. And that it also allows us to distinguish the tumor cells from the normal cells. And so, as I mentioned, we can get a much better understanding of the on-target and off-target effects of these drugs. And also, with the high-throughput instruments such as the iQue screeners, we can minimize the sample requirement, also reduce our cost, and maximize the results that we get from our analyses. And also, I just want to thank my many collaborators. This project would not be possible as a single group project. It is really a multi-group effort and also a core service effort and a clinical effort. And just to thank my collaborators. And also, if I didn't point them out earlier, the members of my group, Heikki, Mamun, Maya, and Deb, who have really spearheaded the establishment of multi-parameter analysis in our laboratory. And also, I'd like to thank the many patients who have donated their samples as well as our funders. So thank you for your attention, and I look forward to getting some questions a little later.
Excellent. Thanks very much, Caroline. That was a great presentation. You provided our audience with some great information on new high-throughput drug discovery assay methods and how the iQue screener can assist investigators with their experimental design. Before beginning Catherine's presentation, I want to remind everyone once again to submit questions for our Q&A session at the end of the presentations. Simply type your question into the Q&A box on the left-hand side of your screen and hit submit. And we'll try to get to as many questions as we can. All right, Catherine, the floor is yours.
Well, thank you very much for the introduction, and also thank you for the invitation to present today. At the 2014, AstraZeneca acquired two iQue Screener HD that we placed into our global high-throughput screening center based near Manchester in the United Kingdom. But a year later, we also acquired, our colleagues in Sweden acquired iQue Screener Plus, which is based in Gothenburg, and this one is aligned more locally to the cardiovascular and metabolic disease, but also the respiratory and autoimmune disease area. These instruments are therefore supporting a very wide and broad portfolio of assays and activity. Today, though, I would like to focus on our HTS assay, and in particular, I would like to present to you two different screens that we have run on the iQue Screener HD. The first one is a cell-based assay that we run in a 384 well format, and the second one is a bead-based assay that we run in a 1536 well format. And I will go into the details of each of these assays and the pros and cons that we've had with both of these. So our first assay, as I said, is a cell-based assay, and this is an assay this is a screen that we run for amyotrophic lateral sclerosis, or ALS. This is a fatal neurodegenerative disease. It leads to muscular paralysis and ultimately death. We use yeast cells because these have been shown to be a very good model for ALS. They show a phenotypic characteristic that are associated with the disease, and in particular, they show the mislocalization of the target, the aggregation and the toxicity associated with the target. So for this screen, we work with two different targets that are linked to ALS, fuzz and a repeat of an extranucleotide, which is PR50. And those targets are toxic to the cells. So for this assay, we needed something that would allow us to measure cell growth or toxicity, so cell number. Traditionally, people working with yeast cells will use absorbance to monitor cell growth. This is a very easy and low-cost assay, and you just measure absorbance at 600 nanometers, so the turbidity of the culture if you want. But we had several targets that we wanted to investigate, and what we wanted to do was to multiplex and run two screens at once. And for that reason, we decided to investigate the use of our iQues. So what we've discovered actually when we started developing the assay is the iQue is actually more sensitive and more accurate in determining, getting the proper cell number, as can be seen on those two graphs at the top of the slide. What we've done is a serial dilution of cells and then measure in what is depicted in the black line, or what we expected, which is depicted in the red line. And as you can see on the left-hand side, this is the absorbance measurements, and on the right-hand side, this is the iQue measurements. And what you can see is, as I said, the iQue is much more sensitive than the absorbance and more accurate in its cell number, as after a certain while in absorbance, it doesn't make that much of a difference. The number of cells that you have, you get the same absorbance. So we decided that the iQue was suitable for this assay. What we did was we obviously monitored the growth curve of the yeast cells so that we could pick a time point that was suitable for the assay. Those yeast cells that we used, we had two different strains, one per target, so one for fuzz, one for PR50. And those were made by constructing them under the promoter, galactose promoter. So we could control the expression of the target themselves. So if we grow the yeast into a media in glucose, using glucose as the sugar, we wouldn't have any expression of the target, therefore the cells will grow. If we swap to a media that contains galactose, then the target will start being expressed and the toxicity will be seen. So this is how we conducted the assay, by just monitoring the growth of those yeast cells into a media with galactose as the source of sugar. But as I said also, because we were willing to multiplex those two, we had to first show that multiplexing the two different strains were not going to be detrimental to our hit identification and to the data quality that we could generate using the iQue. So what we did for that purpose is to multiplex the cells, we labeled one of the strains. So what you can see on that slide is the basic principle of the flow cytometer. So as everybody knows, you just have your sample is getting sucked into the detector and carried to the detector with the sheath fluid. And what I've depicted here is basically how we gated our different population. So we run this assay in a 384 well plate format as I stipulated. So what we did is we added beads that were unlabeled beads, so then we could identify every single well of the plate. And what you can see in the central graph is basically the scatter plot of the two populations that we had. So the small one on the top is the beads, the bigger one is the cells. But from the cell population, this comprises the two different yeast strains that we were dealing with. So as depicted on the right-hand side of the slide, you can represent different ways for judging the labeling of your cells. So you can do a histogram, which is the top plot, where you can see that one of the strain is giving us a cell population at a low fluorescence, and the other strain is giving us a cell population at a higher fluorescence. At that stage, I should emphasize that yeast cells are inherently fluorescent, so they have an autofluorescence anyway, so which is why you might be surprised to see that even the unstained population is still giving us a reading in a fluorescence channel. Also, because this assay is just monitoring yeast growth after a certain time in media, we expected that not all the compounds will rescue from the toxicity induced from the expression of the target. So therefore, we might have some yeast cells that would divide and grow, but we might also have some cells that will not grow once the target was expressed, as these will be toxic. Which is why, again, depicted in a different way on the middle panel in the dot plot, you can see the two different populations. So the unlabeled cells will remain at a constant fluorescent level. They're not labeled. Their fluorescence is not going to change over time. While the labeled cells, because some of them will divide when we have some compound that will rescue the toxicity, those cells will divide, and therefore their fluorescence will change over time, which is why you have two subgroups, if you want, of that same population. You have the parental cells that have not divided, and you have the daughter cells that have divided and have a lower fluorescence. So you can also see this in the bottom graph, where you can see if you plot your graph in terms of fluorescence on two different channels, you can clearly see the two different strains that you have in your well, one being the unlabeled one and one being the labeled cells. As I said, because we multiplex, we first had to prove that by multiplexing, we wouldn't be changing the quality of the data and we wouldn't be affecting the hit identification of our screen. So what you're looking at on that slide is to ensure that we were not affecting anything, we run a set of around 15,000 compounds, and we run them either on each strain individually or in the multiplex setup. So therefore, what is plotted on the two graphs on the top are the activity seen in a multiplex condition versus the activity seen in a standalone, so single population conditions. As you can see, those data are pretty much very well correlated, and therefore we can see that we have no effect of the multiplexing on the identification of the hit for each of the different strains. Also, what we do is we run those validation sets on two separate occasions, and we plot the data in a blend-Altman plot, which is the bottom plot that you can see on that slide. What this is depicting is the average of the activity on the two different occasions on the x-axis versus the difference between the two sets of activity on those two different occasions. What you're looking for here is a set of data that will be centered on 0, as if your assay is 100% reproducible. Your activity will be identical on the two different occasions that you've tested your compound. You also want to see a minimal variability around the average. So as you can see in those blend-Altman, for instance, this is indeed centered on 0, and the two other horizontal lines are depicting two standard deviations from the average. So what you can see here is we have an assay that is reproducible and with very tight standard deviation. So, the way we started running this assay is we use our iQue screeners, and we had our cells in the plates. So the protocol required that not only we shake the plates at the beginning of the assay, but also during the reading of the plate, because the cells will sediment during the readout. So if you don't shake your plate, obviously your cell number will go down as you read your plate from left to right or top to bottom. And And this is, again, we have to keep in mind, this is flow cytometry. And this is yeast cells. So yeast cells are known to be sticky, and we're dealing with targets that are toxic, so will induce cell death. We will have cellular debris into our sample. Those samples will go to the detector. And flow cytometry is really relying on a very clear path for the fluidics. And anything that you would have like that, such as cell debris, would result into a blockage. And the blockage of your path is depicted in that slide. So basically what you're looking at on the top right will be your perfect trace. So what you're looking is the x-axis is the time and the y-axis is the event. So every single vertical blue line represents one well of the plate. And what you want to see is every single line is equally spaced versus time, as the sipping time is constant for your samples. And also what you see in the gap in between is the time when we had to wash during the plate readout and also shaking the plate. So at the beginning, as I said, our protocol was set up so that we had to shake the plate while reading every 48 wells to prevent cell from sedimenting, but we also had to clean during that time. So every 48 wells we had a quick clean of the system to make sure that we got rid of any possible debris that would block the fluidic path. And what you can see on the left is what happened when you have such a fluidic instability. So what you can see is instead of being nicely spaced out, you have some lines that are missing in the time. And if you don't do anything at that stage, if you let your system run like that, then you go to the middle graph where basically your path is blocked and you can't detect anything. The system can't find the wells because there is a blockage in the way between the sample and the detector. But what we found is we were in constant, during our screen, we were in constant talk with IntelliCyt, and they suggested that we change the buffer. And we changed our buffer to an IntelliCyt buffer called QSOL. And what that has done is actually it has really helped our screen, and it has really helped the readout, because what we could get away with now is not having to clean during the read of a plate. You might say, you know, why is that so important? Well, we'll put it into context. We have run 1,400 plates for that screen. So every single time that we can shave off a read of a plate is something that is important to us, as this means that we can increase our throughput. So if we can read a plate in 10 minutes instead of 20 minutes, it makes a huge difference. So running with that buffer has clearly improved, as you can see, going from left to right. You've got a really good trace now. I would still recommend, though, that when you run a cell assay, you still monitor the data live so you can see what's happening during your screen. So assay, what we've done is we've identified quite a few compound that would be specific to certain targets or would be pan-target, but we've been able to run it as a multiplex assay because we use the iQue screener. As depicted on those graphs, I've just shown you a few examples of, for instance, a compound that would be specific to one target rather than the other two, as can be seen in the two graphs on the top. Or, at the two graph at the bottom, a compound that will be hitting 2 targets out of 3, or a compound that will be hitting all targets altogether. It's very likely that those set of compounds that will hit all the targets with the same activity will be a non-specific compound, and it's more likely that it will hit the expression of the target, so it will interfere, for instance, with the promoter, and therefore will give us the same activity regardless of the target. But a compound that will be hitting 1 or 2 targets will be pretty specific to that target. And currently we are progressing those hits through a panel of cascade assays that have been put in place by our collaborators. So, changing tack, this is the second part of my talk, and what I would like to present now is a very different set of assays. It's a bead-based assay. And this is an assay for GTPase, RAC1, and this is an assay that we run in a 1536 well format. At the beginning of 2016, AstraZeneca entered a partnership model with IntelliCyt. Thereby, we relied on IntelliCyt know-how and expertise in flow cytometry, and we, as AstraZeneca, brought up the biology. What we wanted was, we came up with assays that we would like to see, and IntelliCyt, we developed a proof of concept for the assay. So this assay that I'm presenting now is such an assay. This is an assay for RAC1 that IntelliCyt developed for us. This is based on previous literature. So what we have for this assay are beads that are coated with glutathione, and we come up with our target RAC1, which is tagged with GST. So your GST tag will bind to the glutathione on the beads, and what we can do now is have a binding assay of GTP, fluorescently labeled GTP, called body P-GTP, and we can test for compound that will disrupt the binding of the GTP to the target. So what we would be monitoring is basically the beads and the fluorescence associated with those beads. So in the bottom left panel, what you can see is the first proof of concept. If you don't coat your beads with anything, you don't have any fluorescence label. So that proves that actually the binding of the GTP is specific to the presence of the target on the beads. And you can bring back down that fluorescence level by treating the beads with unlabeled GTP, therefore competing off the labeled GTP. And this is depicted in the pink and purple line at the bottom versus the control with the labeled GTP, the green line on the top. It is also depicted on the right-hand side, which is now looking at a flow trace, where you can see you have your control without any label, then your label with the GTP, body P labeled GTP, and then you can break that back down to basal level when you use unlabeled GTP. So this being put in place, step was to miniaturize the assay. So this was important to us as well, because by moving from 384 to 1536, that means we can decrease the volume of the assay. And if we decrease the volume of the assay, we decrease the cost of the screen. So by moving from 384 to 1536, we move from a 15 microliter volume to a 6 microliter volume. And what you see here are the final steps that IntelliCyt put in place to come up with the protocol with, again for us, as I stipulated before, it is always a compromise between reading time and data quality. So we want the best data quality, but the fastest reading time. So we try to find the smallest SIP time that we can use to ensure we have the same data quality, but with a smaller, shorter reading time for the plate. So this is the final protocol as we run it. We monitor, as I said, the beads, the singlet of the beads, and the fluorescence associated with the beads. Again, before running any screen, we always validate them. So we use a set of compounds, 7,000 compounds, that we run, as I explained for the previous screen, that we run on 2 separate occasions, and then we plot the blend Altman, where we plot the average of the activity on those 2 different occasions versus the difference of the activity on those 2 different occasions. And again, you're looking for something that is centered on 0 with a very tight horizontal line, very tight standard deviation. And this is exactly what we had. But we also, for this assay, we also monitored the IC50 of unlabeled GTP, and during the screen we could see that it came up regularly around 150 nanomolar. So while we run that screen, what we found is around 4,500 hits. So 4,500 molecule out of our 500,000 compound that we screen that would interfere with the binding of GTP to the RAC1. Those hits were further profiled into our cascade against two related GTPase, RHO-A and CDC42, as we wanted to identify RAC1 selective compounds. And those three curves show you three different compounds that are selective with various level of selectivity. As you can see, the bottom one has some margin for RAC1 versus the other two, while the top two are clean against RAC1 and do not hit the other two targets. All those compounds that we've identified are coming through a variety of structures, and we are currently looking into further activity of those compounds, and those compounds are in the hands of our collaborators in France. So when we talked about flow cytometry, people always ask me, how long does that take? Flow cytometry has a reputation for being slow and low throughput. So basically, this is what we found. So when we run in a normal working hours mode, what I mean is we spend an hour in the morning setting up the iQue, preparing the iQue, so that's cleaning, QC of the iQue, and then we run non-stop, and I really do mean non-stop, 7 hours on the iQue. We run plates after plates. We have the iQue automated. And if we do it this way, we can run 11,000 data points, so 11,000 wells, if you want, per day, per iQue system. So a 500,000 compound screen will take you 4 to 7 weeks, depending on the number of iQues you have at your disposal. As I said, if it's a cell assay, I would recommend that you keep monitoring the data as they go live so that you can intervene if something happens. If you've got a blockage in the system, you can intervene, clean the system for 5 minutes, and rescue your run. If you don't do anything, you're going to have a blockage and you're going to lose all the subsequent plates. But we anticipate that if we run on a 24-hour cycle, so if our iQues are constantly running, we can do 35,000 wells per day, per iQue. So the same 500,000 compound screen will now take us 2 to 3 weeks to achieve. So far, last year in 2016, we've run over 2 million data points on our iQues. So what have we learned from running these screens? As I said, flow cytometry is sensitive, but it's also susceptible to blockage. So you have to be careful with the samples that you put through, as any blockage will reduce your sampling and will lead to poor well identification. So that's the reason why, again, I emphasize that it's important that you keep monitoring your data as they go through so you can check that everything is running fine. But I would also encourage you to try different buffers, because what we found in our hands is changing the buffer has greatly improved the robustness of our run and has allowed us to also simplify our protocol. As I said, we got rid of the cleaning that we had mid-plate. So then we reduced the reading time of a plate from 18 minutes to 11 minutes, which means a huge increase in our throughput per day. When we run the 1536 assay with beads, we saw different problems. So basically, we never had any issues with fluidic instability. We never had any of this. The sample was really clean. All we had was beads and buffer. There was no debris here. But what we saw is, because we were using beads in a 1536 plate, we had to shake those plates quite strongly. And what we found is the plate alignment can be ever so slightly shifting. So you have to consider this and check your calibration regularly. It's only like 2 minutes, but I would recommend you do this. And also, keep track of what you're doing, and also put in place a strong, strict routine for cleaning your iQues every single day and stick to it. It's really important. It's 20 minutes. It makes all the difference when you run a screen on that scale. Finally, I would like to finish by acknowledging all my colleagues who have worked with me on those screens, but also our external partner. Jane Shorter, the University of Pennsylvania, and Aaron Gutler at Stanford in San Francisco, who have generated the yeast for us and are part of the ALS screen. And Vincent Sozo et Gervaise Laurent in France at the Institut du Thorax that are working with us on the GTPase screen. Thank you very much for your attention, and I'm looking forward to your questions. Thank you.
That was a great presentation. Thanks, Catherine. I think our audience now has a greater understanding of how the iQue screener can streamline cell-based and bead-based assays during the drug discovery process. So before we start the Q&A session, I want to let everyone know that it's your final chance to submit questions for our speakers. We've already received a bunch of questions, and they look great, but I want to encourage the audience to keep them coming. All right, so it looks like we've got some really great questions that have already started to roll in. So let's go ahead and begin the Q&A session. Bear with us for a few moments while we get everything squared away, and we'll get started. All right, everyone, so let's get to our first question. Our first question is for Caroline. Caroline, one of our audience members would like to know, they say, you mentioned that the iQue HD and the iQue Plus are in high demand. Do they look at other diseases as well? Interesting to see the biobank 5 fresh sample viability. Do you routinely test the biobank over set time periods?
All right, that's a great question. So we do have several groups at FIMM and other nearby institutes that are utilizing the instruments for other diseases. So these might be solid cancers, for example, and also immunologically based diseases. And so they're primarily using fresh tissue and just doing a dissociation to isolate the cells that they're interested in and then analyzing them on the iQue. And then for our hematology biobank samples, yes, we do routinely test the viability over time of the cryopreserved cells, at least 2 times a year. We haven't used the iQue instruments yet for this analysis, but yeah, that's something that we definitely should do.
Great. Thanks very much, Caroline. Our next question is for Catherine. Catherine, one of our audience members would like to know, have you tested the effect of the QSOL on cell response profiles or the general happiness of the cells?
Yes, we've done that. So basically, maybe I'm sorry I didn't explain clearly. This buffer was not added to the media of the cell, so the cells were not grown in that buffer. The buffer was only used on the buffer station that is placed in the iQue. And yes, we did test that. So we run the system in parallel where we were using our normal buffer, or then we moved to that buffer, and we just said that indeed it had absolutely no detrimental effect on the cells themselves, on the profile that we were seeing, on the compound that we could identify. So yes, we tested extensively, and it had absolutely no effect whatsoever on the cells.
All right, thank you, Catherine. Caroline, another question for you. Can you comment more specifically on any successes your ex vivo drug testing has had on patients in the clinic, in particular on relapsed patients? Caroline, are you on mute?
Yes, sorry, I am here now. But yes, so another great question. We do have some published results on relapsed AML, and what we have seen with this disease, they're usually using the results for patients who really are quite far along in their disease. There's really no good treatment options for these patients. And the only drugs that have been available are those that are already approved. They haven't been able to access for clinically investigational drugs. But what we have found is that about maybe 40% of these really acute patients do respond quite well to the drugs that are used for them, and I think we would probably see a much better result if we did have access to some of the newer drugs where we do see very good results in the ex vivo assay, but haven't been able to access the drugs for in vivo use.
All righty, thank you very much, Caroline. Catherine, another question for you. Audience member wants to know, was I correct in hearing that you have been able to achieve or beat IntelliCyt speed specifications to read plate, aka do less than the advertised 12 minutes for a 384 weight? Also, how many plates did AstraZeneca screen in 2016?
Well, I was not aware that there was a competition for the reading time, so if we've defeated IntelliCyt themselves, then have we won an IQue? So yes, indeed. So if 12 minutes is your generated reading time for a plate, yes, we achieve a faster reading time than this. And that, again, I would say, this is something that is based on the assay. So you're going to have to play around and just see what are the best conditions for your assay for what you are looking at. So it's always, as I specified, it's always a compromise for us between the reading time, which is very important when we run at scale, and the quality of the assay. So we always try to find the best condition. So yes, indeed, we were able to run a 384 plate in less than 12 minutes. In terms of the number of plates that we've run quite a few. So the cellular assay, we run over 1,400 plates as a primary, and after that we run all the concentration response, so that adds quite a few plates on top. The bead-based assay was over 400 plates, and again followed with the concentration response. So I think in total we've run quite a lot of plates on our IQue so far, and we're still running. So we're not trying to achieve the maximum number of plates run on the platform, but we've generated over 2 million data points, so that's over 2 million wells if you want. So that's quite a lot. So we've used them extensively. We've really put them through their paces.
All right, thank you, Catherine. Caroline, one more question for you. If you use patient immunotype to gate your data, what is your strategy when the immunotype of the patient is not yet available or is fully defined? And in what fraction of your AML samples is this true?
So we have really just started applying the multiplex assay to our AML study. And basically, for all patients, we're still running our single parameter cell titer glow assay for the 525 different drugs. And then going back and taking cryopreserved cells from some of the select patients where we do have very clearly defined immunophenotype data from the clinic to do subsequent more in-depth analysis. But of course, I think one of our goals is, to analyze these samples fresh and real time with the iQue screener, so we can get a much better idea of both on-target and off-target effects of these drugs.
Great, thank you. And I think this will be our last question overall. This one's for Catherine. Catherine, and maybe actually even Caroline could chime in as well. What is each speaker using for automation of iQue, orbiter versus other robotic arms?
Right, so I'll take that one first, Caroline, if you don't mind. So we currently have our platform automated on a high-res biosolution system called the Assay. So we have 2 iQues that are fed through a robotic arm. We will move the system to a bigger platform, which is still part of the high-res biosolution that we're putting together in collaboration with high-res. So this is the system we're using. So this is not an orbiter. This is a high-res biosolution platform.
And from our side, we haven't progressed to complementing our iQue with a robotic arm yet, but hopefully some point in the future.
All right, great. Thanks, guys. And with that, we've come to the end of our webinar. So I want to remind everyone that the webinar will be archived on our site at www.genengnews.com for up to 1 year. So if you missed it, or if you want to watch it again or want to forward it to any of your friends or colleagues, which we always recommend, you could do that. I'd like to thank Caroline and Catherine again for their informative presentations, and I'd like to thank the audience for their attention and thoughtful questions. And a very special thanks to IntelliCyt for sponsoring this webinar. Hopefully we'll see you all again at another Gen webinar in the near future. Goodbye for now.